Hello, merit microscopy students. This is Dr. Georgie, and today we're going to talk about one of my favorite, mega favorite actually, topics, the scientific method. Basically, how do we know all this? By all this, I mean the kind of things we talk about in science classes. How did we figure it out? It was always other humans that did the work. And you have probably heard about the scientific method in another science class at some point, and it's described this way. First, you make an observation, notice the world around us, then pose a question, probably based on that observation, and then propose a hypothesis. A hypothesis is basically a potential explanation for what you observed for the answer to the question. And in order to do science on it, you have to come up with some sort of testable predictions in that hypothesis. So, and otherwise it's just a thought exercise, which is wonderful and a lot of fun actually. But to do science, so to speak, now you've got to have some way to test it using the tools that we have in science, including, of course, microscopy. So at this point, you design experiments, which is a lot of fun. And then you do your experiments, and then you're collecting data and doing the experiments. This is what happened. So you're recording it in your lab notebook. And then you're going to draw a conclusion, which is, hopefully a yes or no to your question, to your hypothesis. And the conclusion is an opinion. It's an informed opinion, right? And you're going to start over with new questions because every question answered leads to more questions that you can ask. And I'm pretty sure you've come across this explanation of the scientific method. And I hope you have. It's a good way to describe what we do. And it's kind of mostly true. Let me put it this way, all parts of these are true and important parts that are part of the scientific method. But one thing that gets lost in the translation a lot is how we actually do science as opposed to how we report it. Let me show you with this drawing. So when we present info we being scientists, we present info out about what we've done. We present it in a very linear fashion: observation, hypothesis, experiments, data, conclusions. And it looks like we're all these brilliant geniuses because we always get an answer and an interpretable good, an answer we can interpret that's solid with good proof. And here's a very strong conclusion. Well, that's because we wrote it that way. Okay, we had all the data, we did the journey, and then we present it that way because that's just the way we present things. This is why I highly recommend going to talks, because in a talk you will sometimes get the real story behind the story, because how it happens in real life is more like this maze of arrows, and sometimes you're just going off for months on a turns out to be useless sidetrack, and then maybe the sidetrack is actually sometimes what it ends up being the useful thing. Sometimes, to be honest, we get data and then we ask a question around it because cool things happen and our instinct says, let's do this. And we often retrofit the hypothesis. So when we write the paper, we're going to ask the question such a way as so that the answer is yes. Does this molecule do this? Yes. Well, we probably started out with some other idea about what it might do. And unless it's really bizarre what we found, we'll just say, oh, that's what we were going to do all along, because it just makes it a simpler story to tell. What we're really looking for as scientists is, what is our data? Okay, so the framing is nice, but it's just framing. So don't get fooled by the fact that when you read a paper, it's like, oh yeah, look here, everything went perfectly as expected. No, I guarantee you nothing went perfectly as expected. Real life science is messy and complicated and full of surprises and intriguing. It's very much like, it's a real life detective story. Well, we're trying to find out what the truth is in life. We're trying to get answers to our questions, and the process of doing so is not linear. There's a lot of loops and iterations and muddling and getting lost and wait, let's try to make sense of this. Let's see what's going on. So creativity plays a huge part in science, and you don't necessarily get that from the way that it's represented in the media, or the way we ourselves report out what we're doing. Like I said, if you go to a talk, instead of reading the paper, the talk, they'll sometimes tell you the interesting journey to get to the data. You can also ask people at conferences at posters, what their journey was like, and they'll tell you the whole thing. The printed report is just that. The paper is a report. It's here's the basics. um, so a lot of textbooks talk about Alexander Fleming as an example of, the scientific method. And if you pay attention to the story, you'll see that there was not so much creativity in his case, but there was a huge element of chance. Chance is the other thing that plays a huge part in science and scientific discovery. We can approach every question with all the rigor we want, and the best of tools that are possible at that point in time. But unless chance favors us, um, it's just not going to happen. There's so much work and research that never gets published because it's all the things we try that don't work out. And I think most scientists I know who are more on the basic science, where we're doing pure experiment, we really are kind of going over the edge of knowledge and trying to find the next handhold to, a fact somewhere out in that ocean. Most people will say they spend 90 to 99% of their time doing stuff that does not result in any sort of concrete answers or proof or story that you can put together. So, so Alexander Fleming's story is kind of fun because it shows that good fortune, but it also shows another element that is really important to be a scientist, and that is stubbornness. So, and a certain amount of self-confidence, I guess you can call it. So he was not studying mold. He was a bad science. Well, he was bad at being able to grow bacteria, which is what he was trying to do. Talk about side routes that led somewhere. Mold was a side route for him. He basically wasn't able to grow a dish of bacteria. I mean, how hard is it to grow a dish of bacteria? I'm always trying not to. But in his case, and this is true for many people, it's even easier to grow mold on that dish of bacteria because mold liked the same stuff. So he had a contaminated dish of bacteria. And this is the point where I think self-perception is really important. Instead of just saying, oh, well, I'm a terrible scientist, I can't even grow bacteria, I only get mold, I should just go get a different job. He said, well, I seem to be good at studying mold. Okay, I'm studying mold now. I'm good at growing it. I'm going to study it. Okay, so Alexander Fleming says basically, I am now studying mold. And then he goes on to discover penicillin. Oh, because he got curious about the fact that the bacteria seemed to sort of be repelled by the mold. There was this zone of nothing in between the bacteria and the mold. And then he thought, this was his hypothesis and it was correct, maybe the molds are secreting something that kills the bacteria near them. In fact, they were. It was penicillin, from the name of the mold that he happened to be growing accidentally. And that was the first antibiotic. And it was literally life-changing because our life expectancy between antibiotics and understanding hygiene, basically washing our hands, gave us an extra 30 years of lifespan. So, thanks science. And now I laugh as I say this because now, of course, we return to the importance of washing our hands. But back then at the turn of the century, it wasn't really known or understood. We were just figuring out bacteria. Remember, scopes just got good enough to even see these things. And it was a little crazy to say there's these random things on you that you can't see, but they might kill you. Well, turns out it's true. Okay, so curiosity is really important in science. Creativity is really important. Chance, good luck. And, having some good self-regard and just saying, I'm not, having that moment where you go, instead of going, oh, I'm terrible, I can't grow bacteria, I can grow only mold, get curious and go, wait, this is interesting. Mold is interesting. Let me see what's going on with the mold and start asking questions. And then you can, basically add 30 years of life to everybody, to a lot of people on the planet. So science is about the search for truth. Honesty is really important to us, integrity. There's a lot of things that you can do in science. You can be kind of immature and bratty, for instance. If you're a good scientist, people will forgive you. But if you're not honest, no, you're not a scientist anymore. That's game over. We're all searching for the truth, and we have to really be interested in the truth, right? The truth of what is happening around us. So we're investigating reality. Not all questions are appropriate for science. I feel like sometimes there's sort of pop science or the edges of science where people ask silly things like, there's a lot of overhyped stuff. Like, for instance, I would say that does somebody not love me is not a science question. What is the meaning of my existence? Religion, philosophy can answer that, even literature. But science is more about investigating concrete things in the physical world. And I know there's some people put too much stock in certain hormones and so on and overinterpret them. But when you dig, you'll realize the reality of hormones is beautiful, but also more complex than just like, oh, a hormone goes up and I have certain feelings. Anyway, coming back to what is science and where I was sort of saying sometimes people overuse it in boundaries and places where it's asked. You got to look at the questions that can be really asked by science. Science tries to look for a cause and effect as much as it can, but it can also be descriptive. So science is the naming and organizing of species and and families and phylogeny. Basically, people looked around the world and said, cats, they're all a species, and dogs are all a different species. Yeah, they have 4 legs and hang out with us, but they're different. Things like that. So description is really important, sometimes underrated, because good description is still important. It's still happening, and it's where everything starts. It's where we make the observations. We do like our science to be able to predict things. So it's a test of science if we can take things that we've seen happen and then put it to work and predict, based on what we know, we're going to be able to see XYZ effect or something like that. And of course, I think we all know that there's a lot of logic in science. We love logic, this follows this and there's rules on it. But in truth, most good scientists I know also use a fair dose of gut intuition. And you can say that's the creative side, if you will. And just a little quick aside, in case you haven't already encountered this yet, biology uses the word theory very differently than we use it in the rest of the English language. So if I say, I have a theory about whether you'll show up to the appointment on time, or I have a theory as to whether you'll wear a mask when I see you or something like that, that means I'm speculating, I'm guessing. And so this creates a lot of confusion, because when a biologist says there's a theory about something, they're using it as scientific jargon, as the language of biology. And in biology, by theory, we mean we know it rock solid as life ever gets, as humans ever figure anything out. So there's always, I like to say, there's always an asterisk in science. Like, we can test and prove everything, as much as we can. And, there's always a tiny bit of doubt, but sometimes the doubt is so tiny, really, really, this is true as humans ever figure anything out. And a theory is something, it's a set of knowledge that is true. So it's not even just one little fact. It's a whole set of knowledge. For instance, the evolutionary theory, which of course, is subject to that misinterpretation. There's a lot of other theories too, like the cell theory. Life is organized into cells. So less controversial, but equally true. Okay, so When we think about science, we're digging around, asking questions, and coming up with answers. How do we know when we've come up with the actual answer? And to put it more in terms of science, what is enough proof? What is enough proof? When can we say we know it as opposed to it might be so, or I wonder if it's this way? When have we actually proven it? So let's look at another of these known things in biology, these theories. Life comes from life. It was very poetic and beautiful and profound if you think about it. And um, basically, in biology, this is what we know now, that if you want to get something alive, you have to have something else alive, and you can make more. But the original spark of life, if you will, the genesis of life happened a long time ago. And ever since then, it's been a continuous stretch of living things that just through evolution, differentiate off into different species or cell types. There's a lot of variety. Nature loves variety. But this thing that we call life is continuous. So, like, for instance, as a biologist, when I hear people in talking pro or against abortion, um, ask, when does life begin? That's nonsense to me as a biologist. Life doesn't begin with each human. Life begins a long, 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 long time ago, and then it's a continuous, unbroken chain of life ever since then. You're really saying, when do we give this organism human rights and so on, legal rights? But when do its legal rights begin? But its life began a long time ago. It's just a life comes from life. I bring this up just to give you an example about proof, because we didn't always know that life came from life. For a while, until quite recently, until the 1800s, we thought maybe dead organic stuff could give rise to life. And one of the favorite ways of proving this that people had was to say, well, look at meat. Like, we all agree that meat, we're eating meat, it's not alive, it's taken out of the organism, it's even cooked. Well, really, they were looking at raw meat at the time. But, so here's the meat. And yet, there's this thing called spontaneous generation, where although this thing is an object or dead, spontaneously, the spark of life just reignites randomly all over the place. You just never know when you're going to get life exploding out of nowhere and nothing, and starting from scratch. And as opposed to what now we think, happened a long time ago with some RNA and the primordial soup and all of that, molecules getting together. Okay, so in the 1800s, they said this was happening all the time. In fact, just look at meat. You can leave some meat out there and just let it sit there, and days, months later, you get a new life form appearing, these white little wiggly worms called maggots. So look, maggots have they weren't cows or ducks or whatever you were eating. You're not getting new ducks, but you're getting new little white things that we now know are maggots. Okay, so a scientist named Francisco Redi decided, okay, I'm going to disprove this. I don't buy this, because here's what I noticed. Here's the observation. He said, oh, I see a lot of flies around, house flies around that meat. I think that those maggots are the baby version, the juvenile version of those flies. I think the flies are laying eggs, and the eggs hatch, and then you get maggots, and the maggots turn into flies. And he said, I don't think there's no such thing as spontaneous generation. Turns out it was true. But I pick on this example because it's in a lot of textbooks. But the classic example has him, how's he going to prove this? Okay, he sees flies. He's like, I think they're the cause of life. I don't think the meat is giving rise to meat. So how's he going to prove this? He's going to leave some meat out, just leaving it out as usual, and expect to see maggots. And then he's going to cut off the axis for the flies to meat. So he's got let's say, 2 jars, and in one there's some meat, and he leaves it out overnight. and he comes back to see maggots the next day. The second jar, he actually covers it with cheesecloth, which is just this, lets the air through, but it's the flies can't get to the meat. And it comes back, it's on the same windowsill, he leaves it out, comes back the next day, no maggots. So 2 jars, one had meat and now has maggots. The other jar was covered, kind of covered, semi-covered, and has no maggots. That's his evidence. And from that, he's going to say this huge conclusion where, oh my gosh, I think that there is no such thing as spontaneous generation. Life comes from life. Now, peel back the fact that you probably believe he's correct, which I do too. We all think it now, but realize that this original experiment, it was more than 2, but it was actually just a couple of jars. And I'm going to take a second to let you think about this question. How many jars should he have tested? I think our instinct goes 2 jars, really? So should he have tested 3,017 jars? Would that prove it? If half of them, had cheesecloth and no access for flies, and the other half, had just a jar with meat and, oh, look, maggots, would that be more convincing? Of course. Of course, it would be more convincing if he replicated this. So where is the line? If you'll accept 3,017 jars as proof, but not 2, how about 53 jars? How about if he used 323 jars? It's not so simple, right? If you had a statistic class recently, you probably know the answer, but just off of the top of our heads, I hope this has gotten you going, yeah. When did he do enough to really prove it? And then also think about the way I told you the story. Did he really prove that flies cause maggots? Or did he just show that if you cover something with cheesecloth, you don't get maggots? Because nowhere in the story did I tell you, if you replay it, nowhere did I tell you that flies were observed in the actual experiment, right? So hopefully your skeptical mind is already firing. Always pick apart everything people say to you, and then, put it back together for social amenities. But if it's science, pick it apart. Okay, anything anybody says, be skeptical. And so you should have, I hope some of you may have caught that I didn't ever mention any record of, like, we're blaming the flies, but were they even there? And what proof do we have that they were there? These days we'd set up a grad student and a camera and they'd review the recording data. Back in then, for all we know, he went to sleep all night and he just woke up and said, oh, I'm right. Look, one jar has maggots, the other doesn't. So if you're going to accuse the flies, so to speak, make sure you have proof that you saw them. And then start thinking about what else could have explained his results, where an open jar has maggots. Presumably there were flies spotted near, and a closed jar doesn't have any, basically. Maybe the flies were hovering around, but they can't get through the cheesecloth to lay their eggs. As a microscopist, I'd want to see the eggs under a scope. Show me there's eggs. Show me there aren't eggs in this other meat. I'd love to see them, follow the life cycle and see a maggot turn into a housefly. What about the temperature? What about the location of where he did the experiment? Is this true only in his house, or is it true in other houses or labs? So these are all variables. Hopefully, once you start thinking about them, you can think of a lot of variables. What if somebody came along during the night, switched the jars as a prank? That's a variable. Well, a lot of science is figuring out what are the reasonable variables. So if you live in a town full of pranksters, maybe that's a reasonable variable. If you live miles away from anyone else, probably unlikely. Can you explain what happened? Maggots in some jars and not the others. Can you explain the facts by blaming Martians, for instance? The Martians come down and plant maggots only, and they only like open jars for some reason. Well, do you have to, it's called controlling. Do you have to control for that variable? Most reasonable people would be like, we haven't proven Martians exist, so we doubt they come down to interfere with your experiment. Um, and what I'm getting at is proof depends on the question you're asking, and proof depends on the conditions of the experiment. Proof depends on what we already know. We're all writing a giant story together as scientists. We're all chipping in a piece of the story using the techniques that we've got, using the knowledge we have so far. And this is why we do peer review for publications, is we have other people, other scientists look at your experiment, look at your data, and then look at your conclusions and say, almost always do more experiments when you send in a paper, and they're almost always going to say, do more controls, actually. So scientists check each other out. Basically, we look, and we're very skeptical. Where is this? Sometimes I've seen people think we're hostile. We're not hostile at all. We enjoy this process. I get very excited if somebody points out a hole in my research. I'm like, thank you. Oh my gosh, I'm so excited. I didn't see that. Let me go test it. So we're all trying to arrive at the truth together, and we're all looking over each other's data. That's why you have to present the data. You can't just present the conclusions. The paper is there for me to go look and go like, did you do enough controls? Okay, you did. You convinced me. When a lot of us are convinced, then, and there's a lot of data, then we can maybe even call it a theory if it's really important. So what are these controls I keep talking about as being so important? So how do we prove things? There's two fundamental ways. One is that you reproduce it over and over again. So other people may have done that same experiment over and over again, but we would get nowhere if that's all we could do, right? We'd all still be doing the same experiment or the same 5 experiments. Again, in the general sort of how to put this, like in TV and books and the way that non-scientists talk about science, you would think that that's the only way we have of proving things is, can we repeat stuff? Is it reproducible? That's a minor way that, sure, we do that too. If we must, we reproduce things. Mostly we're trying to do new things. And so the way that we prove things is by setting up the experiment with proper controls. So it's when you design your experiment to internally prove the answer. So within your experiment, you're going to get the truth out because your experiment will sort of self-report out the truth. Mostly what you're doing is if you're looking for a cause and effect, you're asking the question, what else could cause that? Is there another possible cause? In Reddy's case with the jars and the meat, we see flies, and then we see the flies can't get to the covered jars. So we see flies landing on the meat and presumably laying their eggs. Is there another variable? Is there another species of animal that's doing this? Is there another reasonable variable? So we're not going to worry about proving the Martians didn't do it, but was it the flies, or are there birds also jumping into the meat, or, I don't know, worms or something else? So controls are about eliminating other possible answers, other possible explanations. And I'm going to give you a homespun example to understand controls. If you relate to this, great. And if you don't worry about it. It's just an example, which is why it's in orange here. It's a little story called Rodolfo and the Mystery Yeast. So here we go. So my good friend Rodolfo calls me at 2 a.m. my time in a panic. Oh my gosh, there was a flood in my bakery. Rodolfo now lives in Austria, but stayed with me a few years ago. And Rodolfo's like, there's a flood and everything's out and everything's damaged. And I have my special yeast starter that makes my bread so good that I developed when I was in California. Can you please, please tell me that you have some of it left. And I'm like, dude, I don't make bread. Like, how would I have your yeast? And he's like, well, when I was staying with you, I used your kitchen and I probably froze some in your freezer. Can you go look? Um, and so I find some powder in a Ziploc bag that may have been labeled back then, but the label's worn away. I'm not sure what it is. I don't really tend to have powder and Ziploc baggies in my freezer. So I think it's his yeast. But am I going to tell him that? And he's going to I need to prove him because he's going to be so excited if I found it. So I need to prove it. And to prove it, I'm going to make some bread. And let's say there's just two parts to making bread mix and yeast. Okay. So I have some white powder that I don't know what it is. I think it's yeast. I hope it's yeast. I'm going to add it to bread mix and I'm going to put it in the oven and I'm just, skipping over all the steps, but I'm going to hope to get some delicious bread. If it's not yeast, if it's just, I don't know, salt you might have put in there or something else, I don't know, then I'm going to get crackers, right? The bread won't rise. So I'm going to take the packet and I'm going to try to bake a loaf of bread. But Rodolfo's really counting on my answer. And what if it doesn't work? Do I know for sure that wasn't yeast? Think about it. What might be another explanation? Let's say, okay, packet of white powder, whitish yellowish powder, and bread mix, and I try to bake bread and doesn't work. If you were Rodolfo, what would your first question be to me if I was telling you that? Like, yeah, I found some powder in a baggie, but I don't think it's yeast because I didn't get any bread from it. I'm going to give you a second to think. What would be your question? Be impertinent because you're a scientist now. Be a skeptic. Probably the first question should be, Giselle, do you know how to make bread? Did you even turn on the oven? I don't trust you. I don't trust that this is Rodolfo. I don't trust that you know what you're doing. Like you say, you got nothing. Well, how do I know? That doesn't prove anything to me. I know who I'm dealing with. That's why I was like being pertinent. It's fine. Almost always, one of the first things we do is, does a scientist know what they're doing? Ask if, do I trust the scientists? Do they seem to know what they're doing? So it's fine. I need to prove myself because that's science. So to prove myself, just because I know Rodolfo would ask me this, and so would I if I were him, I went out and got some known yeast and I baked in parallel a loaf of bread, and it's beautiful. And it shows that, look, I can bake bread. Here's some known yeast. Here's the bread mix. My oven works. I know what I'm doing. Yes, I'm sleep deprived. Thanks, Rodolfo. But I was still able to, with known yeast, bake some bread. And so as a contrast to my answer to him of no, this baggie is not yeast, I am saying, but look, here's a beautiful loaf. So bread is possible. You can, make bread in my kitchen. I can make bread in my kitchen, should I really choose to do so. Now, what if, on the other hand, I do have a loaf of bread come out and, my result is a positive result? Yeah, I get a loaf of bread. I would still want to have what's called a negative control side by side in parallel, which is just the bread mix without yeast, so that I can prove that the magic ingredient that made the bread rise was indeed that yellowish white powder from the freezer. And so, since I don't bake, it's probably his yeast. Of course, we could always use a microscope too in this example, but that's another thing. So I hope what you're seeing, let's see if it will go forward. I used to clap. Oh, well, it's not clapping because we're on Zoom here. I'll do my own clapping. A tale of good controls. So what I hope you're hearing is that I had to prove that I can bake bread. That's a positive control. Look, I can do this. I can get this result. And then I also had to prove that I can also not get the result. That's called a negative control. And so anytime you're asking a question, you run side by side with your question, your experimental um, run, you run other experiments side by side at the same time, basically in the same place, that are expected to give you a positive result and a negative result. And based on that, you can then make a conclusion about what you're actually testing. And so we do this in all fields of science. And depending on what your field is, the controls, the positive and negative controls will be different things depending on your question, but they're there. Anytime you're doing something, they're there. And those of you in clinical labs know that you always run control runs to show the machine is working that day. And so you can trust the results. Otherwise, how do you know that the machine isn't wonky and you're going to get a bunch of positives just because the machine is contaminated or something? So this is done at all levels of science, and it's very important to always have good controls. We live for controls. So here is some text around that. So if you're doing an experiment, you're looking for a result. You're looking for something to happen, or sometimes it's for something not to happen. But you're hoping for something different to happen, and that is caused, or sometimes not, by the variable that you're testing. And when I say data, results, and facts, it's all the same thing. It's things that actually happen that you're going to report. And the controls are designed so that your result is specific to the cause that you tested and not to other variables, not to other reasonable variables. So if you start thinking about all the variables that could have given you the result of, for instance, bread got baked, you could start coming up with some silly things like the Martian snuck into my kitchen and planted a beautiful fresh hot loaf of bread there. Well, is that likely? No, based on what we know, I don't think I need to run a control against Martians because I don't know how to do it either. I don't know how to prove there weren't Martians, but maybe I could set up a camera that records the whole thing and you can see me putting it into the oven, coming back, and so on. So anyway, we look for reasonable other explanations and then we rule them out. So a negative control shows that you can get no effect, that it is possible to get a no answer, if you will. And it's especially useful when your experimental run did give an effect. So this is me showing that I can in fact get no bread because what I'm trying to get is bread in the instance of having bread. A positive control shows that you can get an effect. So this is me going out and getting yeast and making sure I can show you that I can get bread. Now, if with the experimental run, I got bread, this is less exciting. Ideally, I ran just bread mix with nothing in it. And, it was crackers. And I ran bread mix with the known yeast in it. And then I also ran bread mix with the unknown in it. And that way, whatever direction the unknown goes in, I can interpret those results. So if I get crackers, I know that, yes, I can make bread. If I get bread, I know that, yes, I can make crackers. Okay. So ideally, you should have always a positive control and a negative control alongside your experimental variable, your experimental run. Now, in real life, you may have several positive controls and several negative controls. This is where the art of science comes in, is figuring out which controls and how many and how often and so on. Sometimes it's just not possible to have a negative control or positive control depending on the situation. We're going to go into some examples in a little bit, and hopefully you'll see this. All right. So if you thought about this before, maybe because you're already working in a clinical lab, the idea of controls is probably second nature to you. If you haven't thought about it, in your brain, it feels like it's doing backflips and twisting right now, and which is a negative control, which one's a positive control, then please take some time with this, think about it, research it online, look up COVID test and controls, because that's a whole intriguing subject there. I'm going to try not to get off on that track too much, but ask me about it during the live Zooms. So here we go. So controls is really how we figure things out in science. And to be honest, it's our least one thing to do, because it's the thing that anytime like you go to the head of the lab, they're like, if you're in grad school, your PI, principal investigator, she's going to say, oh, do more controls. Nobody ever says do more cool, fun experiments. They always say do more controls. You haven't proven it yet. And when you submit a paper, they're like, more controls, please. So as scientists, we know they're super important, but we're also a little sick of them, because they're the part that we know the answer. We want to do the experiment where we don't know the answer, the part of the experiment where there's no known answer. Sometimes we'll do what's called quick and dirty experiments, where we don't do the controls, just to see if it's possibly gonna even work. And then we go back and we do the controls. So that's part of the messy, like, zigzag of science. It's not as linear as we reported out. Okay, so research design is also known as experimental design, and it's basically two parts: how to answer your question and how well have you proved your answer. So how are you going to answer this question? It's basically dependent on, is it answerable? Do we have the tools and technology and knowledge set up so that we can start asking and answering this question? And how well have you proved your answer is, have you done enough controls to satisfy the reviewers, basically, and everyone else? Anybody that you show this to, are they like, yeah, you've proved this? Or are they like, no, I'm not so sure you proved it yet? By anybody, I mean other scientists in the field who understand what you're doing. How to answer your question. There's a joke that a lot of scientists like. So I'll share it. It's silly and it's used, you may have heard it already, but there's two friends, it's late at night, and they're just out walking. And these days they both have masks on. They may be 6 feet apart. But one of them starts cursing and the other one says, hey, what happened? Oh, I opened my wallet to put something in, and a $20 bill fell out. And I can't find it. It's so dark out here. And so friend number one says, okay, well, I'll help you look for it, and walks down the block to the end of the block. And friend number two is like, yo, I lost it over here. Like, why are you going to the end of the block to look for it? And friend number one, the scientist, says, well, because there's more light over here. So the street is better lit over here and I can look here. That's how science feels a lot of the time. There's a lot of things we want to answer, but we don't have the technology to answer. And so we have to go where there is light, where there is the technology. And these days, to be honest, the technology is accelerating at such an exciting speed that the amount of questions and the kinds of questions we can answer it's just phenomenal. Like I said before, it's a really good time to be in science, because a lot of things are becoming possible and we're getting a lot of exciting answers and developing exciting techniques all the time to ask even more questions. Okay, so just a few little things about science. You might hear, often people talk about this, that there are two types of data: qualitative, which are observations, and quantitative, which are measurements with numbers to them. So it's true, and we do tend to prefer quantitative whenever possible. If we can put a number on it, we're a lot happier. Numbers are pretty objective. We all perceive numbers the same way. So 6 feet apart is 6 feet apart. We can measure it, for instance. It's not, it's not subject to who's saying it, not in reality, not in science. There's ways that we can measure things. Qualitative observations are still important, and there's an art to doing them well. Tools. A lot of times when we are doing clinical trials, which I'm going to talk about in a little bit, we run something known as double-blind experiments. That is basically an experiment where So you're giving you have doctors and nurses give somebody new medicines, and um, both parties are blind. The person who is taking the medicine doesn't know if they're getting the medicine or the placebo, which now you know is a negative control. It's not supposed to work. So it's just a sugar pill, for instance, or salt water or something like that. Um, so the person doesn't know whether they're getting the thing being tested or not. They've signed up to test out stuff, but half of them aren't going to get placebo. And the health person administering it also doesn't know. So there's no giveaway. And the idea is because we're humans, we're gonna, if the doctor, let's say, or the nurse seems really excited to give me this, I'm like, oh, I bet I got the one that really works. So nobody who's administering the experiment, if you will, really knows. There's codes, and they're opened after when the clinical trials have happened. They're opened, and then the analysis is done, and there's less basically human bias in it. Is the idea. Interestingly enough, what we have observed with humans, there's a very strong placebo effect. So just the act of going to, let's say, a doctor and getting some sort of treatment ends up making people better, even if the treatment they're being given is just sham treatment, not a real treatment. This is a very interesting thing when you do experiments with humans. It seems to be less of an issue with mice and rats. They're not as excited. They're not like, oh, here comes the scientist with, my daily treatment. I'm going to feel better. So they have different opinions. In fact, there's a really great um, example of how, like, we figured out that stress can lead to clogged arteries because the experimenter, who was like sort of the head of the inventor of this field, was really bad with mice. Another scientist who had bad hands. His name is Hanselli, and this is described in How the Why Zebras Don't Get Ulcers by Bob Sapolsky. This amazing book. And the scientist kept dropping the mice he was trying to inject for another entirely different experiment. And there would be like a half-hour chase around the lab every time he was trying to handle the mice. And these poor little mice ended up with completely clogged arteries, which is where we figured out stress leads to clogged arteries. Okay, so I'm sorry, I got off on a tangent there, but it's one of my favorite science stories. So coming back to, so anyway, we do double-blind experiments with humans when we're doing clinical trials. Sometimes maybe we should be doing them with mice. It just depends. And then statistics. Statistics are our best friend because, like when I was asking you how many jars did Reddy need to do to prove it to you. Thank God statistics has an answer to those kind of questions. Otherwise, we would just spin our wheels forever. So math is just a huge friend to science in all sorts of ways. It's really an integral part of it. Types of experiments. You might hear in vivo from, I think it's Latin. Yes, Latin means in organisms. It's always like in italics like that. In vitro means in glass, in other words, in a lab. Nowadays we have in silico, in silicon, which means done on a computer. And we do try when we're developing new drugs to start with cell models, so cell lines, so in vitro, that's considered in vitro. And then in actual animal models, while still in a lab, that's in vivo, in the whole organism and whole creature. And then if we're talking about, let's say, oh, I don't know, a vaccine, it's 2021, or, any other new drug. We go into clinical trials. I get a kick out of the fact that you've all heard of this. I used to have to explain it. And although if you're in the health field, you've heard of a clinical trial, but now everybody's heard of them, right? A clinical trial is when we we test something, a potential medical treatment on humans. And we always start with phase 1, which is a small amount of quite healthy human volunteers, and we're just making sure that it's safe. So all kudos to these brave people who are willing to be, I think, heroes for the rest of us. They're perfectly healthy and strong, and they tend to be young. And they sign up and they're told, like no human before has ever taken this treatment, whatever it is, a new drug or anything. But are you willing to be the first small group of humans to do it? And we're just looking to see that it doesn't negatively impact their health. Phase two is when you've passed phase one, and then you start recruiting for the target population for that drug, and you're testing for efficacy. In other words, does it work? Does it actually do anything now that we know it's safe? And you're still testing for safety always, but your focus and your design is now now you're going to ask the question, does it work? So if it works and it's safe, then you scale up to phase three, which has a large number. Like with COVID, the vaccines was 40,000 participants, a huge number statistically, if you think about it. And you're looking for both safety and efficacy. You're kind of rolling it out now. You're doing a third run of testing. It's all the same testing, but you're looking at the data really carefully. And if it passes the trial, it goes to the FDA for approval. And then it can be dispensed by doctors or nurse practitioners or naturopaths, anybody who prescribes medicines. Phase four, the FDA still keeps its eye on this medicine or vaccine or whatever. And after that, so separately, the FDA is always going to monitor that. And so health data is turned in anonymously to the government, to the FDA, so it can kind of track things. As you'll see, that's really important. Phase four is also if a company decides to test it for other uses. So it's post-approval testing. So you may have heard like these mRNA vaccines were rushed, blah, blah, blah. They went through all these phases. And in fact, it was some of the best clinical trials ever run. So it wasn't that they were rushed, it's that we actually had been doing mRNA vaccine work for 10 years. And we got incredibly lucky, talk about luck, oh my God, that the vaccines against COVID did seem to work. Because guess what? We've been working on SARS and MERS, we being scientists, for a while with not much luck. So there was no reason to think that the new SARS-CoV-2 vaccines would work, but thank heavens almighty, they do. And not only do they work, but they work really well. And they went through phase 1, 2, 3 testing. In fact, more rigorous, larger numbers, better run clinical trials than ever. It's that usually things take a while and it's done very slowly. Like you slowly recruit the people and you slowly wait for the analysis. And then when it goes to the FDA, it can die for 2 years before you get an answer because the FDA is understaffed and tiny. So everything was organized. So everything was go, 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 ready to go. Like, let's start recruiting phase 3 people if phase 1 and 2 turns out to work. If it doesn't, well, then nothing will happen. So we typically don't do that with a new drug. We're like, let's do phase 1, let's take our time, let's analyze it. Okay, yeah, it looks good. Now let's see, can we find some other people for phase 2? Okay, let's take 6 months to enroll people. Oh, a year? Oh yeah, sure, a year to enroll people. It's just that it was all hands on deck. Let's do this better, efficiently. This is really important. Not that we skipped any steps, we just did them better and quickly. Okay, so speaking of clinical trials, for instance, I was sort of not saying this in the previous thing, but in the 1990s, there was this huge scandal where they realized that all clinical trials were done on males in their 20s, mostly pretty much all white males in their 20s, because guess what? That's where that's literally the people who testified in front of Congress about this said they intentionally excluded women and they did not know where else to find anyone else. Like, for instance, I don't know, somebody Black or Latino or something other than a white male. Why? Because they were med schools and doctors and researchers at kind of elite institutions. And the people who were willing to be, especially in phase 1 and 2, were typically college sophomores. Literally sophomores, not freshmen. They're still too new to college, not juniors. They're like, I'm over this. But sophomores would like answer these calls was somebody willing to volunteer? So bless their hearts that they were willing to volunteer, but shame on the researchers who didn't know where to find other people. And the reason, it was called minorities back in the day. The reason that there were very few women is because they said, well, women could be pregnant, and that scares us. There are things, and this is true, that alter the embryo that don't alter the woman, and we don't want to deal with those complications. We don't want deformed embryos because of our research. And in these congressional hearings, there were people who were like, have you heard of a pregnancy test? I don't know. Do you think you could administer a pregnancy test? Do you not trust pregnancy tests? And they were kind of like, oh, good idea. It's a lot of men who were running these clinical trials. Okay, so this was pointed out to the scientist, and um, I'm laughing because it's, these biases. Like, we like to think we're all about logic and so on, but then you look at really glaring biases like this, where people are just like, yeah, but I don't want to deal with a woman being pregnant. Good heavens, how could I deal with that? Oh, I don't know, really simply. So the the regulations say that you have to find a representative sample, and there's an interesting history of this if you want to look into it. It's good and bad. It's like, then they had to find people who weren't white sophomore male students, and there's some good things that happened around that and some less good things, and it's still to this day. But these COVID trials were amazingly representative. They did a good job of going into different populations, different places, different ages. Another thing that was a big deal, especially with COVID, and that's sort of unusual, is they found people of all age groups, including skewed older, average age in the 50s, because obviously this is more deadly to people who are older. So that's who they wanted to test the vaccine on. The idea of informed consent came about because scientists and doctors used to be able to just test on anybody without telling them what they were up to. And so now there's some very good legal standards saying you need to explain what you're doing, and people need to sign off and say that they understand what they're doing. And if you can explain it well, it's on you, because you got it. That's informed consent. Basically, you'll learn about Henrietta Lacks, or if you don't, look up the first cell line. There's some really interesting history around that where her family said, you took her cells and you didn't give her credit or ask her or tell her, or, like, that's not okay with us. So they put scientists on check and said, hey, we care. We care what is happening to the body parts of people, and we need to have some say in it. And interestingly enough, there's also, okay, so there's the Tuskegee syphilis study. And I was saying, interestingly enough, this has been in the news lately. Usually when I teach this class, people are like, the what? Unless, depending on who you are and your background. But so you might have heard of the Tuskegee Airmen, which is Black people who were amazing aviators. There's also, same city, in Tuskegee, there was a really evil doctor, was super racist, super evil, who was conducting a syphilis study. And the thing is, so syphilis is a brutal disease that makes you also go, it's just a very horrible way to die. And you go kind of crazy. It's obviously, it's an STD, so sexually transmitted disease. And the good news is that antibiotics, actually, I think penicillin, no, is it? Anyway, antibiotics are a quick, thanks, Alexander Fleming, and your curiosity. Antibiotics work really well with syphilis. And so it's not a threat to us anymore. It's still around, y'all, but it literally is completely vanquished by antibiotics. You just take antibiotics for two days, and instead of dying a horrible, gruesome, long death, you actually, you're fine. It's no biggie. If you have access to the drugs, of course. And that's where this syphilis study comes in, because we figured this out in the 40s. A lot of soldiers were getting it, notably. But anyway, and so there was an army base, and they were, this horrible doctor whose name I have chosen to forget, was experimenting on Black soldiers, and intentionally studying them because he wanted to learn about the physiology of dying and, how people died from this death. And that is beyond unethical because he was doing this well into the 1970s, y'all. 1970s. This study and this doctor and his testing went on for decades after there was a known cure. And he was withholding the cure from his patients just so he could study and watch them die, basically. So you don't get more chillingly racist and evil than that, right? And you might say, well, he was clearly super evil. But the thing is, nobody caught him and stopped him for the longest time. And there were a lot of people around him. And so what were they doing? And he even had some nurses who actually whistleblowed on him, who were Black, who were working for him and were uncomfortable with this and were kind of realizing that he was withholding treatment from the men who did end up dying, several hundred men. So this is a horrifying story, a very American story. This is us, you guys. This is the US. Let's face it, we're good, we're bad, we're complicated. We do horrible things like this, and there's always a bad egg or two. But the thing is, this wasn't a bad egg. It was a lot of people enabling this guy who were letting him do it until finally somebody shut him down. Because of this famous study, this and other things propelled this legal notion of informed consent. So we did get informed consent out of it at the expense of a lot of people. I find it interesting that it's in the news lately because it's like that racist tricks will never end. People are now saying that African Americans are not willing to get the COVID vaccine because they know about the Tuskegee syphilis story and they don't trust doctors. And it's like, well, they shouldn't trust doctors. However, actual data and actual research has been done on this. And first of all, African Americans are saying no to the vaccine at the same rate as everyone else. And second, most those who are saying no because they don't trust doctors are saying no because they don't trust doctors now, not from the 70s. Because there's still a lot of, I shouldn't laugh, this is serious, but it's so absurd sometimes how people will twist arguments around. And it's like, white guilt is saying like, oh yeah, I don't know how, the convolutions are crazy around this kind of repurposing of the Tuskegee syphilis study to blame Blacks if we don't get herd immunity. It's just, the creativity is all in the wrong direction here. So anyway, so you may have heard of it. One thing you probably haven't heard of lately is Vioxx. It's one of my favorite stories. I may have alluded to it at some point earlier in this semester because it was the first drug approved under new rules that are still somewhat true, where the FDA everybody was like, the FDA is taking too long to approve new drugs. And so let me see when this was. This was maybe 10 years ago or maybe 15 at this point. So it's been a while, but this is still happening. So it's true, the FDA is super slow except for the COVID vaccine, which was put at the front of the line, as it should have been. Instead of just saying, let's hire more scientists to be on the FDA review panels for clinical trials, the solution was, let's just let the drug companies themselves supply all the data instead of having outside companies do the clinical trials. Let's just trust the drug companies and the pharmaceuticals to provide the data. This is such a bad idea. And basically a lot of people said that the FDA changed from being a government regulatory agency of the pharmaceuticals to being seen as like, they're like, the companies are our clients. We have to keep them happy. Otherwise we won't have a job. And this still needs fixing, y'all. So Vioxx came out, and it was also the first drug that was in this country targeted directly towards patients in the marketing. So it was marketed straight to patients. So in 1 year, an enormous amount of people took Vioxx because it was the new painkiller. And then people died by heart attacks and there were lawsuits and money was, paid out to the victims and so on. And Vioxx was pulled off the market pretty quickly, thanks to one person who was doing post-approval review at the FDA. At the time, there were only 4 people doing post-approval review, even though think of how many hundreds and hundreds of drugs are there. And this guy managed, he's a hero, he was a whistleblower, he managed to catch the fact that Vioxx was causing heart attacks and killing people because it was so blatant. But here's now, any of that can happen because sometimes, as good as our clinical trials are, you don't know until lots of people take a drug and maybe you didn't catch something and biology is complicated. Sure. Why was all this money paid out? Why the pharmaceutical companies guilty? Because here's the key thing in this whole story. It was a totally predictable side effect. It was a known pathway that in our bodies, a known metabolic pathway that we understand really well, that it was affecting molecules that, are responsible for pain, but they're also responsible for the clotting response in our bodies. So it was entirely logical, predictable, knowable that this drug as a side effect might cause heart attacks. It made complete sense. That's why this guy knew to review for that. This is why we want our doctors reporting what happens to their patients, to the government. This is why it was evil of the, and I'm blanking, I think it was Merck, but they all do the same thing. These big pharmaceutical companies do a lot of good, but they also have huge profits and they also make bad judgments because they're profit driven. So they buried the clinical trials data that was showing this was a side effect. And so, are there issues with the way that science and clinical trials are done in this country? Sure, there's some things that are way better, like the clinical trials for COVID were just marvelous in terms of how representative they were, how inclusive, how well run they were. Is there still the chance that people hide data? Yeah, absolutely, when there's money involved. Did that happen with the COVID vaccine trials? Unlikely. Here's the more important thing. The COVID vaccine, there are 0 predictable side effects. Based on all of the science we know, there are no side effects that we can predict. We still went through a lot of testing rigorously just in case. If something does happen later, we really could not have predicted it. This is not Vioxx. Vioxx was totally predictable. A little bit of RNA inside a little bit of fat, it's a miracle it works. It's not predictable that it shouldn't. Okay, let's wrap this up a little bit. I know it's hard. I know it's getting long. It's so hard for me not to talk about science. I love it so much. So what is the process? Labs are little groups. Each lab has its own personality, its own set of rules, its own kind of culture. There's usually the head of a lab who's called a principal investigator in an academic setting, but also in biotech sometimes. That means they're the main person on the grants, but they're really the intellectual lead and they're kind of known for their accomplishments. A lot of times you will see one scientist named. And as I did, I've done it in lectures too, but in reality, it's always a collaboration. It's really one person. It's usually a group. It's usually their lab. Then there's a lot of collaborations between labs themselves. As I said, the literature is really important. We're always reading each other's papers and reports, but there are also oral presentations which tell the fuller story, which are really fun to attend. So posters and talks, and I've talked about peer-reviewed journals. The funding in this country for basic science mostly comes from the National Institutes of Health, so taxpayer money funds most basic science. So that is where you're just asking a question because you're curious, / it's a cool question to ask because you can. It's research that's done in academia and also at some research institutions like the Buck Institute, for instance, I think is NIH funded Institute for Aging over in Marin and other places like that. And then once there's a sort of a way to apply some of the things that come out of basic research, or then it's like, oh, wait, I can do something with this. Then we're talking biotech pharmaceutical or startup companies where they're trying to apply this towards better human health. And let's come back to controls. Let's end with a little bit of thinking about controls. So I gave you the story of bread just as an everyday sort of example, if that helps you think about controls. But let's think about it in a real science context. So let's say I have this question. I have found this new compound, let's call it Z, and does it increase breast cancer growth? Is this, a risk factor for people? It's in the environment. I want to see if it's causing more increased breast cancer. So that's my question. And then how do I turn this into a research project? Well, first, I have to define the question better. So what type of breast cancer? Are we talking tissue culture, mice, humans? And what does increase the growth mean? That's kind of vague. Do I mean recruitment of new tumor cells or proliferation, which is cell division of existing tumor cells? And these are just some of the questions. So, okay, I decided we're going to use a mouse cell line, and I'm going to look for new tumor cells. So conversion of regular cells into tumorigenic cell types. And so I've picked a cell line X, and does this molecule Z, this new exciting molecule, promote conversion into tumorigenic cell types? Here's the experiment. I'm going to add Z to the cells, and then I'm going to count cells that are tumor cells. That is the assay. The counting the tumor cells is called an assay. I should have written that there. It's ASSAY. So can we do it? Can we count tumor cells? The answer is yes, there are many ways. And they mostly involve microscopes, right? So okay, I've got numbers of cells that are marked with tumor markers. Is that more? What does more mean? So I have to compare it to a cell line that I haven't turned into tumor cells. And so that's my negative control, right? Also, do I know for sure that this cell line can turn into tumor cell types? Well, we need a positive control. So let's add in a molecule called Y. Let's say it's, for instance, BPA, bisphenol A, which is something in certain types of plastics, which is being taken out of it, but it's still there, which we know turns breast cells, regular healthy breast cells, into breast tumor cells. So let's run a parallel control. So we're going to have a negative control and a positive control. There we go. We can have several positive controls. We can have variety of negative controls. And in fact, I'll challenge you to think of some, but just to wrap this up, let's think of experimental design in terms of testing a new COVID vaccine in clinical trials. So what are we going to do? We're going to add the vaccine to people, right? And we're going to count the COVID rates. And hopefully here we want to decrease. We want to see that if you get the vaccine, you're less likely to have COVID. You've been following this presumably. So you know that, again, define the question. What's a COVID rate? Is this like death, or is it COVID at all? How do we know? Are we testing these people? Yes, of course, we're doing tests if they're in the clinical trials. What's our assay basically? How are we going to score the level of severity of COVID? Well, that was all worked out. So there is an assay, and people who are enrolled in the clinical trials, that's exactly what happened. You have people, their medical histories, you try to enroll a representative sample of the population in this country, and some people got vaccine, and then you followed them and looked at the COVID rates. So what were the controls? Well, two types of negative controls come right to mind, right? People without the vaccine at all. So, but remember that whole placebo thing. So first of all, people without the vaccine, can we track them? Yes and no. Yes, we have people that we could track and get the data about, right? Because we're somewhat tracking this, not nearly enough. We're not doing as many tests as we should, because the more tests we do, the more knowledge. But we do sort of know what the rates are, especially since people without the vaccine may end up getting sick and showing up in the system. A better negative control, which is what these clinical trials did, is yeah, there's going to be people who enrolled in the clinical trials and they are going to get the placebo. So they're going to get a saline injection. They're going to get the experience of getting a shot, but in that shot, it's just salt water, which is well known to have no effect. If you inject somebody with a tiny bit of salt water, zero effect on health in any way, shape, or form. But of course, the placebo effect, as we know, is strong. Turned out in this study, not so strong. So that was actually good because we wanted it to not, we wanted people not to, like I went to the doctor, I magically am now immune to COVID. That would have been messy. But as it turned out, the rates of a negative control of people who did go in and get an injection, but actually didn't get the vaccine, their rates of getting COVID are kind of the same as the people who never even went in the doors, weren't enrolled in the study. So those two negative controls, rates of getting the disease were pretty comparable. And then what about a positive control? Well, the way to create a positive control is to make sure some people get the virus. So give them the vaccine and challenge it with these are called challenge trials, challenge it with the virus. Nope, we did not do this because it's unethical to do on humans. We did this, we can do this in cells, we can even do this in mice. You can argue about that, but we do at the moment let it happen. But we're not about to go injecting people with a virus to prove that, I mean, that, actually, control the C plus. I just realized I made the slide tonight. It should not say plus the vaccine as I'm saying that. I'm like, why would you do that? That would be a challenge trial. So that is one kind of positive control, is a challenge trial. So virus and vaccine. And it's kind of in between. It's not really a positive control, but it is a control. A real positive control would be just the virus, no vaccine. Okay, so that's the part where we're like, nope, not going to do that. Sadly, a lot of people sort of sign themselves up by just exposing themselves by not wearing masks and not staying away. So there was a lot of data in this country, in certain countries, like this country in Brazil and Italy, my hometown, I mean, my home country, certain countries provided a lot of positive controls, sadly, to compare to the study, which is part of why we know that the studies, the clinical trials were good. And then as a matter of fact, now that we've given it to millions of people, we see the same data, the same statistics, because those clinical trials were so beautifully designed. And I certainly don't say that about all clinical trials, but in this case, things went well in all kinds of ways. So there you have it. There's research design, real life, up to date in our lives, saving lives, using science to save lives. It's pretty phenomenal. I find it really exciting that we were able to do this, that the vaccine, all things considered, is pretty phenomenally good so far, as we know. Okay, so that's it for experimental design, and have a great week.